Fantastic. Yep. So thank you for the introduction. I'm a lecturer and researcher here at the University of Leeds. My background is in medicinal chemistry, and I'm going to talk about some of the work we've been doing in the last 5 years, looking for novel KCNT1 ion channel inhibitors. I don't have any background slides because I think that's been very nicely covered by previous speakers. So we're going to dive straight into the work we've been doing. So I collaborate with my colleague, Dr. Jonathan Lippiatt, who I know has presented at the Foundation before. And he's been working on characterising this channel for a number of years. And this started with a discovery of some initial KCNT1 inhibitors, which was in partnership with Tiffany Therapeutics as part of a PhD programme. And this was published in iScience in 2020. And this is when I came on board as a medicinal chemist and in silico kind of computational chemist as well. We've got a number of arms to this project in terms of repurposing, which I know the Foundation are aware of, so I'm not going to talk about that today. I'm going to focus on the sort of discovery of the novel KCNT1 inhibitors that we've got from a large screen and briefly touch on what we're doing currently with our work. So we have this two-pronged approach, but as I said, for the purpose of time today, I'm going to talk just about the novel small molecules and not talk about the repurposing strategy that we've been working on as well. So a lot of my work, a lot of my background has been developing pipelines for virtual screening. I've been doing this for nearly 20 years now at the University of Leeds, and this was really facilitated by the publication of the structure of the KCNT1 channel in chicken in 2017, and more recently we now have a human structure which was published with a small molecule inhibitor in 2023. So we can take a section of the zinc, which is a commercially available library of small molecules, and we were able to look using our virtual screening platforms to identify small molecules which we predict were going to be able to be pore blockers for the KCNT1 channel. This work was part of an Action Medical Research Grant, which was funded for 2 years and finished last year. And in the initial stages, we purchased 73 compounds from our libraries of 9 million, which we screened. And then we decided to assess these in our cell-based assays. So our first assay is a thallium flux assay. We assessed all our compounds, all 73, for inhibition at 10 micromolar, and we pleasingly found 7 compounds which inhibited our thallium flux at 10 micromolar concentration to the same extent as 100 micromolar treatment of our control compound Bepredil. So our next step was wanting to validate these results using our whole cell patch clamping, which is work that was carried out by John and our researchers here at Leeds. And we were able to see that our molecules did have activity in the patch clamp assays as well as the flex station. We then wanted to look at validating the selectivity of the molecules which we had. And we have a number of compounds which all our compounds exhibited good cell bioavailability in our WST1 assays when tested at 100 micromolar, so our highest concentration. And we did our IC50 determinations again in HEC293 cells. We've got this double kind of heteromeric mutant where we have wild type channels with our Y796H mutants, which we think quite accurately represents what patient populations might look like where they have mixtures of wild type and mutant channels. And we were able to confirm that our molecules still had activity in these follow-up assays, and we also looked to do some whole cell patch clamping looking at the hERG channel, and that's actually the next slide, so we should move forward. We're able to see that a number of our compounds, pointer here, several of our compounds do have effect on hERG. We're definitely not at a stage where we've got our preclinical candidate, but we do have a range of different activities. So Bepredil shows 100% hERG inhibition in our assays at that concentration, but some of our compounds had very low levels of hERG inhibition. And we also developed a panel of related ion channels, and we were able to pleasingly see that a number of our compounds were selective for our KCNT1 inhibition over closely related channels. So we then had some follow-up work done with Cyprotex, who are a contract research organisation here in the University of Leeds, looking at some initial tier 1 ADME. And we can see that our compounds can have sort of sub-micromolar potency on KCNT1 at best. We have a range of different hERG inhibitions. We have a range of solubilities. A lot of the compounds, a lot of our compound problems are with solubility at the moment, which is what we're looking to address. We have some relatively pleasing human liver microsomal stability for some of our series, but we're very highly plasma protein bound, and we were quite pleased with some initial CYP. We don't have complete, we have some quite good lack of CYP inhibition, which we're very pleased with. We realise our compounds have still got a long way to go to be these preclinical candidates. So our current work is working to synthesise analogues of our leads. We're doing this in a data-driven approach. We realise as an academic institute, we can't churn out the number of compounds required in maybe the industry can. So we're using software from a company in the UK called Medichemica, which is an AI-driven tool to aid in designing analogues. So we want to be able to reduce the number of analogues we need to make to improve the properties which we're interested in improving, in this case, very much solubility. So we're using the new structure of KCNT1, which was published in 2023, along with the Medichemica software, our in-house assays, and also in-house synthesis to improve the parameters that we've got and hopefully get a compound to a better stage of development. And we're currently in discussion with a number of companies about partnering with us for further funding applications to the Medical Research Centre in the UK. So to summarise, we've got a number of novel, structurally diverse and synthetically tractable inhibitors of our KCNT1 channel, which we've identified using in silico screening with our in vitro assays. We've shown it's possible to achieve selectivity for this channel versus other structure-related ones, including hERG. We're currently developing our structure-activity relationships to understand our molecules better, and this has been really improved by the publication of an inhibitor-bound cryo-EM structure of KCNT1. So I'm going to finish with just acknowledgements of the people who've done this work. The work I present today has mainly been the team on the bottom of this. I'd like to thank Action Medical Research for funding this, and everyone for listening, and to Ali and everyone at the Foundation for the opportunity to speak today.